Happy Monday, everyone. I hope you had a good weekend. Obviously, I'm sure what's on your mind is the first exam. So we're going to talk a little bit about that. But first, I want to talk about the structure of today. So kind of the first part of today, however long it takes, I have allotted sort of the first hour, but if we're done sooner, then we'll just move on to Unit 1 review. And this is student-led in the sense that I want you to ask me your questions. So take a second to get out your study guide and your notes and think about the things that you have questions about. And then when your questions have sort of started to trickle down, we will do a Kahoot or sooner if nobody has any questions. I do want to also touch base about the unknown ID project. And then when that's kind of squared away, about maybe I'm sort of budgeting about the first half of class for all that stuff. We will spend the last half of class on chapter 6, the growth section. This is part of unit 2, and just to be very clear, not on Thursday's exam. Okay. A quick reminder, of course, that the test is on Thursday and it's going to be proctored on Zoom. So you don't come to campus, and if your lab is normally on Thursday, you must come to lab on Tuesday at the usual time. And for Thursday, what you need is just a stable internet connection and to be able to get onto Zoom with me. We'll talk a little bit later today about what that looks like. Okay, so the first thing we're going to do is just open it up for any questions you have about the first unit. And so that's like anything about the intersection, basic features of microbes, history, the chapter on microscopy. So what is the size of everything? How do you calculate the size of things given a scale bar? How do you use different microscopes? What can you see? How do you calculate resolving power? What does it tell you? What is the importance of oil? All that fun stuff. And then the section on cells. So what are the features of prokaryotic cells and eukaryotic cells, and how are they similar and different? So all that good stuff. If there's anything in that first unit of material that you feel a little bit, oh, and of course, CFUs per square foot hour and CFU per mil. So any math that we've done in the lab, procedures that we did in microscopy that we also covered in class, such as the gram stain and the acid fast stain and all those other special stains, or anything that came up during the Zooms or the video lectures that, that you need to get clarified. What I'm going to do is just have you raise your hand if you do have a question, and then I'll answer them. Sometimes I might draw pictures if necessary, and I'll just give you a second to kind of think about what your questions might be. Brooke, what's your question?
Hi, so I was looking at the study guide, and one of the ones that really kind of stumped me was how does the cell wall structure relate to the O antigen? Because the only thing I really had for O antigen is that O polysaccharide, and that's something to do with E. coli. And I kind of didn't really know what the answer to that one was.
Excellent. So let's go to the study guide. I'm going to share my screen and we're going to look at that question so everyone kind of is on the same page, literally. So this was the one about the, just get there. How does the O antigen relate to the cell wall structure down here? Yeah. Okay. So you're absolutely right. It has to do with the O polysaccharide. So I'll just review that. The gram negative, it's not just E. coli, it's like any gram negative cell has that distinctive cell wall structure, right? Outside of the plasma membrane, there is a thin layer of peptidoglycan. And so I'm going to switch back to the slide so I can kind of diagram this. I'm going to try to switch back and forth here, see if I can. My pictures are going to be very terrible, so just bear with me. So for gram negatives, There is a, obviously here's the plasma membrane. Then there's that thin layer of peptidoglycan that's just loose filaments. It's not connected. There's not a multi-level meshwork like there is with gram positives. And then additionally, outside of that, is another phospholipid bilayer, but its purpose is different than the plasma membrane, which is for exchange. This is for exchange. This additional plasma membrane is for just to be part of the cell wall. So it's outside. And in it, if you were to look at the phospholipid bilayer, you would find lipid A floating around. That's lipid A. And attached to lipid A being a lipid, hopefully that's not super surprising, is something called the O polysaccharide. So I'm going to try to diagram it like a little, so these little lines are like different sugars. So it's just a long chain carbohydrate with different sugars. Some of them are branched, some of them are, different kinds of sugars, and there's lots of sugars. So it's a distinctive, and different species of gram-negative bacteria, their O polysaccharide doesn't always look the same. Like many species have the same O polysaccharide, but you could build this tree lots of ways. And this is part of the cell wall, gram-negative cell wall, not just E. coli, but all gram-negatives. There is an O polysaccharide. It is attached to lipid A, and it can be distinctive. So it's not that it's distinctive to the species. Like you can't look at this and go, that's E. coli. But it is distinctive to the species substrain in terms of that. There might be 17 species that all have this version of the O polysaccharide. We'll call it O1. But then another whole bunch of guys might have something that looks a little bit different. Maybe it looks more like this, that O polysaccharide just has a different appearance, different sugars in a different order. We'll call that O2. And you may remember, and why you're thinking about E. coli is that there's a famous pathogen that's name is it's E. coli. So we know it's in that family or that species, but E. coli has several substrains that have just mutated away from each other just a little bit. And so this one called O157H7 has the 157th version of the O polysaccharide. And that is associated with it also having a particular toxin. It's not that this is what makes it dangerous, but this is kind of associated, that this happens to be on the species that has the toxin. Regular E. coli doesn't have the toxin and has a different O polysaccharide. So it's sort of a coincidence, I guess. It's a little bit like Jeffrey Dahmer's name is Jeffrey Dahmer. Like we know that means he's a bad guy, but he's not a bad guy because of his name. We just know who he is because of his name, if that makes sense. So does that help, Brooke? Does that clarify, or is there anything you wanted me to sort of like further elucidate? Great. Okay. Anyone else have questions about this material? Go ahead and raise your hand if you do. And it might be helpful to go to the study guide and peek at anything that when you are creating your study guide, which is my suggestion, right? And what you should always be doing. And if you didn't do it for this test, you're going to find you wish you had. So make sure you're doing it for the next test. Anything in that study guide that was confusing. Lanisa.
Hi. I can't find it now, and it's bugging me, but last night when I was just sifting through stuff, I saw something about a staining practical. Is that happening, or what
Oh
is
yeah.
that?
Yeah. So, and you're in the Tuesday or Thursday labs usually? Which one?
Thursday morning.
Okay, so I'll see you this Tuesday because you're coming to Tuesday this one time, right?
Right.
And you'll find out more about that. But basically for the next couple of weeks, you have the option, not the option, the opportunity, you must do it at some point, to gram stain any of the species that I provide, and then show me your stain and I'll grade it. So the way that works is the practical window is open for a couple of weeks. So you have several shots at it. You can do it as many as 3 times. If you don't like your first grade, you can do it again. If you don't like that grade, you can do it a 3rd time. It's not a ton of points, it's 5 points. And then what it is you just do the gram stain, you take it up to oil, and then you show me on your microscope, and I grade it like right there and then. I grade it on how well the gram stain went, and then I'll give you feedback so you know if you're going to repeat it, what to try differently. And I also grade your interpretation in terms of did you correctly tell me the morphology, arrangement, and gram status? Does that help?
Yeah. So last week didn't count. My stain was so pretty.
It did not. And yes, so last week was your first shot at it, right? The first time you ever did a gram stain, correct?
Yeah.
And what you were graded on then was, did you do it? Did you submit your picture? And I haven't graded yet because I'm super behind, and sorry guys,
Right.
it's just like been like that. But that picture is in the lab Dropbox now, and that's what I'm going to grade. I'm not going to grade like how awesome you did. I'm just going to see that you did it and that you did all the things I asked you to do, and then you get all the points if you did that. So now you have to take that skill that you've developed and show me that you can replicate it.
Okay. But it's not part of this exam. It's just a different.
That's
thing.
right. It's not, no, because that's going to be an in-person thing that we do in lab together, whereas the test is obviously on Canvas.
Yeah.
So it wouldn't
Cool,
be
thank
very
you.
reasonable for me to go, now go into your kitchen and do a gram stain real quick.
Right. All right. Thanks.
You're welcome. All right. Aminata, what's your question?
Question is question number 3. I don't understand question number 3 on the study guide.
Great. Let me just see if I can get out of here. I'm going to keep that, but introduce a new one. And then I'm going to try to find this. I don't know why when I go to new share, it doesn't show it to me straight away. It's very frustrating, but there we go. Okay. So the third question on the study guide is this one. How do you convert? So is this what you meant? Or do you mean at the bottom?
Sorry, on the staining section.
Oh, in the staining section. Okay.
Yeah.
Let's see. Okay. So 1, 2, 3. Understand the types of dyes and why each is used. Is that the question?
Yes.
Great. Okay. So, so to be more direct, what you need to know about staining is you need to know the steps in the gram stain, the steps in the acid-fast stain, the steps in the endospore stain, and kind of why those particular dyes do what they do, right? So one thing to know is that water-soluble dyes are usually either basic or acidic, and lipid-soluble dyes are not. So do you remember that the acid-fast cells have a waxy coating that normal dyes cannot penetrate? Do you remember that part?
Yes.
Yeah. And so that the reason we have to use the carbofuchsin is because it's lipid soluble and can penetrate those cells, and other dyes wouldn't be able to. So it's kind of getting at that, like, why do we use carbofuchsin? It's also looking at the use of basic versus acidic, although I don't actually, in this current iteration of the class, I don't hammer on this too much, so I wouldn't worry about it, but there actually is a difference between basic and acidic in terms of whether it stains the cell or stains the background. Don't worry about that, but do understand why carbofuchsin is used and why other basic or acidic dyes wouldn't actually stain an acid-fast cell very well because of that waxy coating. So does that part kind of clear?
Yes.
Yeah. And then it is also important that when you're looking at the gram stain or the acid-fast or endospore stain, that you do know that most of the dyes are actually water soluble except for carbofuchsin. It is important that you remember that crystal violet is particularly useful in a gram stain because the Gram's iodine can then thicken it. And the malachite green is particularly useful in an endospore stain because it's really good at getting into things. That's right, Hannah. Yes, the acidic stains the background, the cells repel it. Yeah, and that's something that's part of the capsule stain, though I haven't emphasized it, so I don't actually test on that because people tend to sort of forget, but that's why that's in there. Hannah just commented on that in case you're wondering what I'm talking about in the chat. So does that help you?
Yes.
Awesome. Okay.
Thank you.
You're welcome. All right, so next we have Mai. Mai, what's your question?
Hi, I was just wondering, I did the practice problem on the CFUs for the liquid, and you gave what you would prefer the answer look like for the air. I was just wondering if you could just say, just to make sure, I wrote down what I would have put if it was on the test.
Okay.
Can you just say what you would like us to put?
Yeah, so let's just have a look at, so it was the CFU practice.
No, it was just the one on the study
Oh,
guide.
on the study guide. Got you. Okay, let's go down to that. I wrote that a while ago. Let's see what I said.
Yeah.
Okay, so are you talking about the example or are you talking about this part here?
Let's see. Yes, the UTI one, the example.
All right, so the urine sample was tested for bacteria. Typical CFU is 10,000 CFUs per mil, above 100,000, la, la, la. So then we say, so this volume of this dilution was spread and we got this many colonies. Does this patient have a UTI? So are you asking me like,
I just want to make sure my verbiage is on
Oh,
the
I see. So tell me how you solved this and what you came up with.
So I did 89 times 10.
For the volume? Yep.
Yeah.
And then
I did it times 1,000.
for the dilution.
Yeah. And so
And
I came
then-
up with 890,000
And what were your units?
CFUs per milliliter.
Great. Okay, so that's the way to solve it numerically, and these are the correct units. And that's how to show your work. And you can also say, I mean, as long as it's as clear as this, when I look at it, it's like I can see where all these things came from, but it could be also helpful to say, 10 because of volume,
Okay.
1,000 because of dilution, right?
Okay.
For those of you who are remembering that there's another way to solve this, this also is correct. 0.1 mils times 0.001 is also correct. You'll get the same answer if you do it this way. So if you take the CFUs and divide by the volume times the dilution, the actual fraction, not the 1,000, but the 1 in 1,000, that gives you the same answer. And that also tends to set people up correctly with the units. But then you also want to answer the second question, right? So does this patient have a UTI? What did you decide?
I said yes, because anything over 100,000 was diagnosed as a UTI.
Yeah.
Okay.
Yeah.
I just want to make sure, like, if you were really looking for milliliters in your equation and things like that, I just didn't want to get docked for stuff unnecessarily, if I understand the math.
Totally. And on a test, like if you don't put the units right in here, I forgive that as long as the units are correct here and this made sense. Because I know time is ticking and people are kind of just doing things in a rush. But if you don't use units, that is definitely a deduction. Like if the final answer doesn't have units or there's something wacky in the math or these units are wrong, you can get a deduction, yes.
Okay, great, thanks.
Great question. All right, Melissa, what's your question?
Towards the backside of the study guide, it's under prokaryotic cell structure and function. There's a question: how does gram status correlate to penicillin resistance and why? And I don't quite understand that completely.
Okay, great. So let's start with what you do know. So which gram type is penicillin resistant?
I don't remember.
Okay, great. Let's ask the people in the chat. Let's see who remembers this. Which gram status is penicillin resistant? Right, that's right. So it's gram negative, and the one that is sensitive is gram positives. And then to explain why, let's first ask the class, and then I will draw a picture because I think sometimes the picture can be really helpful in terms of remembering it. But does anyone want to raise your hand and sort of briefly explain what you understand is the reason that gram negatives are penicillin resistant or gram positives or penicillin sensitive, whichever way you want to answer it. Hannah, go ahead.
So the way I remember it was the peptidoglycan bridges or something. It attacks the peptidoglycan cross bridges, and those are in gram positive. So with gram negative, they don't have those. Thus, it's like resistant to the penicillin.
Yes. So let me just switch. That's exactly right. Let me switch to my slides now and just draw that sort of more, some people are more visual and words are not as helpful for them. For those of you for whom words are helpful, that was an excellent explanation. So if we were gram-positive, over here, we've got the plasma membrane, we've got the cross-linked peptidoglycan. It's so frustrating not to have like a legit whiteboard, guys, where I can just draw really fast, so this will take a minute. I think I'm going to try to switch colors for the cross-links so it's more obvious that you can see them. But this is a gram-positive, right? Many layers of peptidoglycan. That's basically what the cell wall is made of. And then the bridges I'm going to diagram here in another color. So they're also cross-linked together, right? You can see these cross-links, and these cross-links are what is attacked by penicillin. So gram-positives are quite sensitive to this because without these cross-links, their whole cell wall basically falls apart. But if we look at the gram-negative cell wall, which I diagrammed in the previous slide, and I'm just going to shoot back to that here for a second. Let's see if I can, let me, yeah, there it is. So you see here that there's just a thin layer of peptidoglycan that's not cross-linked. So there's nothing for the penicillin to attack. Melissa, that was a great question. I'm sure lots of other people had that question. Did that answer your question?
Yes, it's much more clear now. Thank you.
Awesome. Okay. Anyone else have a question that they'd like me to review about anything in the study guide or material that we have covered this section? Kaylee, go ahead.
In the beginning of the study guide, it's the conversions from the nanometers, micrometers. How do I do that conversion? Because I always seem to get messed up on it, and I somehow do it correctly or incorrectly.
Yeah. So yeah, we can go, absolutely. Let's go over that. And I'll try to give you my favorite way to try to remember it when you're under stress. And
Okay.
maybe it'll help you. I don't know. But let's just first just kind of just say what the thing is, and then we'll see if we can find a way to remember it. So if you're going from, well, this is a terrible color. Let me go to like black or something more boring, but easier to see. If you're going from millimeters to micrometers, and you might want to just make yourself a little diagram that's just so you can just refer to it if you can't like logic your way to there. These are smaller, so there's going to be more of them. So whatever that millimeter amount is, you have to multiply by 1,000. Okay, because there's going to be more. Micrometers are more than millimeters, right? Because they're smaller. And if you're going to go to nanometers from micrometers, same deal. These are even smaller. And so there's even more of them because they're even smaller. And then you have to multiply. And if you're going the opposite direction, you have to divide because there's going to be fewer of the large ones. So let's start with that first. Does that make sense?
Yes, that actually makes a lot more sense.
Okay. Now, if you're ever stuck and you're like second guessing yourself, I mean, it's nice to have this. You could just have this. You just bring this to your test and be like, okay, there it is. I'm going to look at it if I need it. But I want you to imagine this. Let's suppose that instead you were converting from meters to millimeters. Because you had the size of a person. Let's say the person was like 1.7 meters tall. And you wanted to know how many millimeters that was. Like you would never be confused by that, right? You would know, of course, I have to multiply by 1,000, right? Because there's just going to be a whole crap ton of millimeters. So if you're going from a large unit to a small unit and you're starting to get confused, or the other way around, right? Then just imagine that the large unit is meters and the smaller one is millimeters. And I think that sometimes can calibrate. Does that help?
Yes, that does help.
Okay, great. Haley's next. What's your question, Haley?
My question isn't a question on the study guide, but rather how the exam is going to look.
Oh,
Is it going to be similar to the quizzes that we take every week after we're done with the material, that you can see every question and that you can flag it and go back to it?
great
Or
question.
is it just like the one question where you have to press next at the bottom where you only see one at a time?
Yeah, that's a great question, Haley, and I'm happy to answer that. So the test is set up one question at a time. So it is a different format than these review quizzes. It's also different in the sense that it's timed, right? That's also a very different experience because those review quizzes have no time limit except for the final deadline. So it's timed for 2 hours. You can only see one question at a time. For the tests where I do it proctored, because sometimes I can't, like, depending on what's happening with the schedule or with a person's makeup test, for instance, if I can't proctor it, then it is going to be locked. And probably you know what that is, right? Where you can see one question and you cannot go back to a previous question.
Mhm.
For the tests that are proctored, you can go back. Okay, so the proctoring part is where you show up in Zoom, you show me your ID, I remind you of the rules, I expect you to follow them. I have a little bit of a keyboard watchdog, so I can find out if people are not following the rules to some degree. Okay, and it's not perfect, I won't catch everything, but I make that explicit. My expectation is that you will honor that. So the rules are that you have to stay within the testing window and not go Googling any answers. But it is open book. You are allowed your notebook, your access to a textbook, you have a calculator, all that stuff. But you're not going to talk to people on the phone or look stuff up on the internet. You're just going to rely on your own personal notes and your own personal knowledge. And then if you were to click out of the testing window, it does flag me. And then I have to do an investigation, which is a pain. So does that kind of, the first part of that, does that help answer your initial question?
Yes. So it isn't locked. You can flag it and go back, but you only look at one question at a time.
That's right.
Okay.
Now, the other thing to know about the test and that I can tell you is the format is pretty consistent, and it is like this. It's kind of a funnel in terms of difficulty. So the harder questions are at the end. Typically, depends on the person, of course, but the first dozen questions are true false. There's a short answer after that that asks you to reflect on the true false questions. There's a bunch of multiple choice and then drop down small multi-part, like 1 or 2 parts sometimes, or matching, or select all questions. And then at the end, the last 6, 7 maybe are longer answer. So let me have multi-part. Some of them are long. They're not long answer, they're short answer with multiple parts, right? So maybe you have to do a calculation and show me your math. Or maybe you have to answer a logic question, like based on what you know, what do you think about this? And some of them are multiple drop down, but they're just longer questions that are auto-graded, and some of them you actually have to write into a text box. Does that help kind of clarify?
That does. Thank you.
Yeah. So guys, the time limit is 2 hours. And for those of you who have any particular specific circumstances, then my ask is that you email me 3 days ahead of time if you're working with CDS or something like that, just to let me know so that I can double check that I've adjusted things correctly for you. But typically the test is 2 hours, and it is longer at the last however many questions do take longer, so you want to save some time for those and some mental energy for those as well. Okay, shoot. Oh, good. All right, Nicole, what's your question? You're still muted, Nicole, in case you are speaking. I don't know if you are speaking.
Oh, sorry.
And before you start, hold on one second. Asked, do people typically use the entire 2 hours? Yeah, they do. Go ahead, Nicole.
I was just wondering if there was going to be a lot of application questions.
Well, so the ones at the end for sure, right? They're application in the sense that they usually require you to understand more than just one concept and do something with it, like work something out, like, solve 4 CFUs per mil and answer a question about that, or, answer something about what if this happened in a gram stain or something like that. So I would say yes, in the end, there are some. And then sometimes the way that I write my true falses and multiple choices, sometimes, even though they're just short to answer, you do have to know more than one thing to answer them. It's not all just like fact-based, like what color is a gram-positive cell is not a question on my test.
Mhm.
But I might ask you, like, if a cell was purple, would you be able to do this with it? Like that kind of thing.
Okay, thank you.
Yeah. Sarah, what's your question?
Is this test on Wednesday or Thursday?
It is on Thursday. I know right now it's showing us Wednesday on
Yeah, because I'm
Canvas.
looking at it. Yeah.
Yeah, but it
Okay.
is Thursday and it will be adjusted. I meant to do that over the weekend. It's just things didn't
Okay, thank
according
you.
to my plan. But yes, it is on Thursday. And and thank you for asking that, because it reminds me to say one other thing. So on Thursday, there's 2 times when you can start it. You do have to start it on time, but you don't have to start it at all at the exact same time. So I'm going to be in the Zoom to officially start the test at 10 o'clock and then later again at 12:20. Okay, so either of those times, because whatever your Thursday looks like, maybe 10 is better for you, maybe 12:20 is better for you. And I open the Zoom half an hour early. So if you were like, I really want to get this test done at 9:30, so you could come at 9:30. What'll happen is you come into the Zoom just like you usually do, but you put your information into the chat and then you wait for me to call on you. And so that can take a little bit of time depending on exactly when you show up and how many other people have gotten there first. By putting yourself into the chat stream, then that's kind of saving yourself that spot in line. And it could be 5 minutes before I get to you. So just come at a time when you're like, I'm ready to start the test and I'm willing to wait a few minutes. Once you're cleared, once we've had our quick chat, I've given you the passcode, I've reminded you of the rules and acknowledged your identification and that it's on my record, then my expectation is you start the test straight away. So like within a couple of minutes, like if you had to go pee all of a sudden, that's fine. But if you've got to go and like, I don't know, like drive to Safeway, maybe not so fine. Hopefully that's helpful. Any other questions, guys? Raise your hand if you have another question. And then we will, Mai, go ahead. What's your question?
I know some of the questions you'll have to actually grade, so we won't know our actual grade for a while. Do you anticipate a time frame of when we will know our grades?
Mm-hmm. So I will tell you the official time frame, and then I'll tell you the realistic one. So officially, I tell people I can turn the tests around in 1 week, but actually in practice, I prioritize the tests over everything else. So it does mean that everything else, like your labs, which are also already behind, get even further behind, but I prioritize the tests because they're so high impact. So typically, you'll know by maybe Sunday.
Okay, great.
Any other questions, guys? The next thing we're going to do is a cell Kahoot, but let's keep the questions rolling as long as you need to. Bimmy, what's on your mind?
So I have 2 questions. The first is, if we get a numerical answer on the exam, what decimal point would you like us to round it to?
Well, that's a great question. So it's like usually 1 decimal place, or even just rounding up to a whole number is usually fine, depending on exactly how exact you have to be for the question. Because usually, like if it was a CFU per square foot hour, there's a couple different ways to solve it. And depending on how you solve it, your answer might be slightly different than someone else's, right? So there's a little bit of leeway there, and I'm not super picky. But with that said, sometimes it's more like I give you some information, then you have to pick a number from the multiple choice. And so then you're going to want to make sure that you are a little bit flexible yourself and just pick the thing that's the closest. Does that make sense?
Yep, thank you.
Yeah, great question.
And then my second question was, I was confused about the significance of the H antigen on the bacterial flagella.
Yeah, so did you follow the O antigen part?
I did, yeah.
Okay, great. So it's very similar. Let's just go back here. And the H antigen could be something that applies to either gram status. It's not necessarily just gram negatives, whereas the O antigen is only gram negatives. So the H antigen has to do with which particular version of flagellin there is coming off of a species. So let's draw a flagella coming off of here, and remember that the protein flagellin is what this flagella is made of. And that protein is kind of, it's the pieces that make up this flagella. I'm just going to draw it as little pieces. They don't really look like this, but one version of this flagellum that would still function as a flagella and be able to assemble looks like this, but another one, a different species, we're going to have species B over here, might have a flagella that's made with a flagellum that's just a little bit different. And so it's got maybe a little like ridge on it or something. And then yet another species might have a flagella or flagella, multiple flagella even, where it's got like a wavy texture. Do you know what I mean? And so this would be like H1. This is the first version of flagellum, and this is H2, and this is H3. So it just refers to the version of the flagellum protein, because small differences in the amino acid sequence can cause different sort of textures on the surface of the flagella that can be detected by antibodies. Similar to the way that there's different versions of the O polysaccharide that are still functional and would also be detectable by antibodies. Does that make sense?
Yep, that sounds great. Thank you.
Any other questions, guys? Alina, go ahead.
So I have a question about prokaryotic cell function. So this is what metabolic process may occur in the prokaryotic cell membrane that don't occur in eukaryotes?
Yes. Okay, let me just screen share that that question from the study guide. Go here. Okay, so that's where is it? Membrane. There we go. Here it is. So this has to do with, remember we talked about the prokaryotic cell membrane and the eukaryotic cell membrane. The similarities between these two membranes being that they're both for the purpose of regulating exchange, right? So ions, sugars, amino acids that need to be brought across the membrane have to be done, that has to be done with transporters. And so the membrane helps to regulate those exchanges and waste and things like that. But there are differences. So for example, prokaryotic cell membranes sometimes also have machinery that you don't see in the cell membrane of a eukaryotic cell. Things like electron transport chains for aerobic respiration or for photosynthesis, photosystems for photosynthesis, ATP synthase. Those are all machines that in a eukaryotic cell are found on the inner membrane or the thylakoid membrane of organelles. Like for instance, in a cell, in a eukaryotic cell, like one of your cells that can do aerobic respiration, part of aerobic respiration is the electron transport chain, and those machines are actually in the inner mitochondrial membrane. Well, that doesn't exist in prokaryotes. They don't have a mitochondria, so they don't have an inner mitochondrial membrane, but they still do have, if they're capable of doing aerobic respiration, and some of them are, they still do have those machines. They're just, they put it in their cell membrane. So that's the explanation. Now, we could dig a little deeper and just say, actually, it's not that the prokaryotes put it there because they don't have the organelles. It's more like that's where it was to begin with, and the organelles used to be bacteria. And so now the inner mitochondrial membrane is the ancestral plasma membrane of the bacteria that it's derived from. So does that help? Or does that make sense?
Yes, thank you.
Yeah. All right. Any other questions, guys? I'm getting queued up for our Kahoot while you think about whether you have anything else that you want to talk about. Okay. So, where is it? So this is the comprehensive cell Kahoot. And then that's the one that's available to you. I believe we've done this one already, but I wanted to run it through again. So let's just quickly do that. So here is the opportunity invitation to join the Kahoot, kahoot.it, or the Kahoot app. And here's the code 6019398. If you want to join in the Cell Kahoot, now is the time. Okay, let's get started. We've got 26 people on the call, 24 logged into Kahoot. That's pretty good. If you have to use the chat or want to join in late, you can do that at any time. The game pin is up here, or you can use the chat. Let's roll. Right, multi-select, which of these are found in all cells? Select all that apply. Yeah, ribosomes and cell membrane, good job. Not cytoskeleton or cell wall. Cytoskeleton is eukaryotic only, cell wall depends on the species. Some do, some don't. Which of these accurately states the structure function relationship? Ribosomes synthesize protein is correct. Multi-select. Which microbes have cell walls? All right, everything except not protozoa. Protozoa are animal-like. Which of these shows the correct cell wall composition? It is bacteria, peptidoglycan. Everything else is incorrect. Which of these are prokaryotes? Bacteria and archaea, correct. Penicillin targets which of the following? If you're just joining in, you can welcome to the Kahoot by putting this pin into the kahoot.it or into your Kahoot app. Welcome. Peptidoglycan crosslinks and gram positives. Is the target of penicillin. Fimbriae, what are they made of? They're made out of pillin, that is correct. Okay, the glycocalyx. Allows some pathogens to evade phagocytosis. Which of these is found only in gram negatives? Lipidae is correct. In E. coli 0157H7, H is for the version of flagellum. True or false? Bacteria can't do photosynthesis or respiration because they don't have chloroplasts and mitochondria. It should be false. Remember, some of them can do these processes, and it's actually those bacteria that their ancestors probably gave rise to the current eukaryotic chloroplasts and mitochondria. Spirochete's corkscrew morphology and movement is due to what? All right, yes, an endoflagella, internal flagellum. Facilitated diffusion differs from simple diffusion. How? Facilitated diffusion requires a protein transporter. Simple diffusion does not. In other ways, they are similar. However, the patient injected with water by accident instead of saline. What's the consequence? All right, true or false? Bacteria dropped in pure water would explode due to osmotic lysis. True or false? It is false. They have a cell wall to protect them. Bacteria often import nutrients like glucose from low to high concentration. What is required? That is active transport. Yes. Streptomycin targets the 30S ribosomal subunit. This antibiotic is likely to affect all bacteria because they all have this structure. C. difficile makes endospores. This explains the persistent nature of the infection because why? Species can survive antibiotic treatment because the spores are resistant to it. Streptococcus pneumoniae makes capsules. This explains the persistent nature of the infection because why? Yeah, that's right. The capsules make the bacterium invisible to your phagocytes. E. coli is a common gram-negative uropathogen. Loss of the pillin gene reduces its pathogenicity because why? That's right. It loses its attachment to the urinary tract. All right, compare prokaryotic and eukaryotic flagella. Eukaryotic flagella have a wave-like motion and prokaryotic flagella rotate is true. True or false? Only eukaryotes have a cytoskeleton. That is true. All right, multi-select. In a eukaryotic cell, the endomembrane system makes all of these except not cytoplasmic proteins. Who proposed endosymbiotic theory? All right, that was Lynn Margulis. This in, well, in the last proximal century. Which of these supports endosymbiotic theory? All of these are lines of evidence that support it. Okay, let's see who takes the who takes the glory today, but I know that you all were right behind these front runners here. Okay, good job, everybody. So let me just go back to the slides, and we're going to take our break right now, and I will come back in 10 minutes. So we're going to be back at 11:05 to start the growth section. And then I'll try to leave a little time at the end for any further questions that come to mind. But for right now, let's take a little break. I'll see you in now 9 minutes. So I'll see you at 11:05.